going to be on gene expression. I'm going to focus on viruses and then bacteria, simply because we know more about those. The next lecture would be on eukaryotic gene expression, which is much more complicated, and we know a little less about it. But we already talked a little bit about gene expression in our last unit, and so we know that it begins with a promoter. So we always have to have a promoter. But what we're going to be looking at here is what determines whether or not a gene gets transcribed or not. So obviously it has to have a promoter, but not all genes are active in all of our cells. Especially if you think of a bacteria cell, bacteria cells are going to adapt to their environment. So if there's something in their environment or not in their environment, different genes will be activated. In us, we're multicellular, so some genes in our bodies are never activated in certain cells and are always activated in other cells, depending on the specific job of that particular cell. And we'll be looking at two different types of proteins, these regulatory proteins, that are going to be controlling gene expression. So we'll be looking at repressor proteins and activator proteins. And I kind of think of the repressor proteins as off switches and the activator proteins as on switches. So we're going to start by looking at viruses. So I'm going to quickly go through the life cycle of a virus. So we start here, and here's our little bacteriophage, and this is our bacteria cell. This is the viral nucleic acid, and this is the bacterial nucleic acid. So the virus has to bind, and it's specific. So this binding is specific to the host cell. So it binds, and it injects its genetic material into the host cell. And that genetic material will ultimately can chop up the host cell's genetic material, stop the processes by the cell, take over the cell, use the cell's machinery to make more viral material, ultimately all the parts that are necessary to make a virus, and then the viruses are spontaneously assembled. They burst out of the cell, destroying the cell in the process, and you can release hundreds of thousands of viruses from this initial one infection. We call this the lytic cycle because it lyses the cell. Some viruses, in us, some DNA viruses, have the ability, instead of going this way, to go over here. And so what they do is they use their genetic material, kind of like a crossing over event, and they incorporate that into the bacterial DNA. And then when the bacteria divides, it copies its genetic material, it copies the viral material as well. And so now we have two bacteria cells that are infected, and those can go on to produce more and more offspring and more daughter cells, and all of those we have viral DNA. Well, now at some point, if the cell's not doing well, not dividing rapidly enough or whatever, the virus can then excise itself out of the host's genome and then go on to enter the lytic cycle. So these are kind of two pathways that some viruses are able to take, and so we're going to look. So a virus may enter the lytic cycle or may go into the lysogenic cycle for a period of time, and what determines that is actually proteins that will bind to the DNA and control which genes are going to be expressed. So if the host cell is doing well and replicating regularly, and there's lots and lots of food available and it's healthy, instead of the virus bursting out of the cell and destroying it, it kind of goes along for the ride and allows the bacteria cell to replicate and copy the genetic material of the virus along with it. So in that case, the CL protein will be produced. The CL protein acts as a promoter for the CL gene, and so it's going to activate and make more of the CL protein. It also is going to activate the lysogeny genes, and so it's going to make the proteins necessary to stay in through this lysogenic cycle. It also will block the CRO promoter, the CRO promoter. So this gets shut down. Now, if the cell's not doing as well, the CRO protein will accumulate, and that is going to activate the lysis genes, and it's going to block the promoter for the CL. And again, it's also a self kind of promoting thing here. So you get more promoter, you get more gene, you get more protein. It's going to cause more of the protein product. So that's kind of how it decides which genes are going to get expressed, and depending on the environment. Another example of how viruses control gene expression is in the HIV virus. So if the HIV virus gets into your cell, so here we have the viral DNA, using the host cell's RNA polymerase, it's going to bind to that and transcribe those genes. Well, as the gene is being transcribed, the host cell has promoters that will bind to that RNA polymerase and block it and stop it. So the virus never actually gets a protein product. However, viruses have adapted to this, and so the viruses can produce this protein called TAT protein. The TAT protein binds to our terminator proteins, right, and stops them, and so RNA polymerase can then activate the gene, transcribe the gene, and then we get the viral mRNA. So that's just another example of how viruses control their genes. We're going to now look at how bacteria control gene expression. So one of the things we see here is there's lots and lots of steps along the way, right? So it's what the DNA actually looks like here, and we'll look at this more with eukaryotes, whether it can be transcribed. Then we have the step of transcription, and then remember we had the processing, then it has to move out to the cytoplasm, and this is a eukaryotic model, I just realized, and then translated, and then and so on. So any step of the way here, the process can be stopped. So we can not make a lot of messenger RNA. We can prevent that from being translated. You can break down the protein once the protein's been made. But the point of my slide here, even though this is not a prokaryotic model, is that the further we go down the ladder here, so we can stop protein product down here, but we've expended all the energy necessary to get here. So it's more energy efficient to stop the process up here than to wait till we get all the way down to here. And so we're going to see how cells stop the process up here. So, all right, one more quick thing and then we'll get into a chemical process. So here we have in prokaryotic genes a certain structure called an operon. We don't see this in our genes, but we see these in prokaryotic genes. So here is a lac operon. So a lac operon consists of basically three things. A promoter, all genes have a promoter, and then downstream from that slightly is an operator, and then after that at least two or more structural genes. And when I say a structural gene, it means that it's going to have a protein product. Some genes code for RNA. We're going to see with the eukaryotic model different types of RNA, but ultimately there's no protein product. So when we use the term structural genes, we mean there's some kind of protein product. And then upstream from our operon, we have regulatory genes. So here's a promoter, and here's a regulatory gene. And these regulatory genes are going to be important in turning on or turning off this particular operon. So here's a chemical pathway. So if glucose is available to an E. coli cell, a bacteria cell, it would rather use the glucose. Lactose can also be used as a food source, but it's not as efficient as glucose. So they'll prefer glucose. However, if there's no glucose, they can use lactose. And if there's a combination, they can use both. But they would rather use the glucose. They're going to get more bang for their buck there. So here is an operon, and upstream from it is our regulatory gene. So here we have the promoter region where the RNA polymerase is going to bind to it. A little bit at the end of that is the operator, and then we have the structural genes. So if there's no lactose, there's not necessary to make the protein product that's used to break down lactose, which is what the lac operon codes for. So this whole gene set would be shut down. So this operon would be shut down. And the way the cell does that is this regulatory gene codes for a protein that acts as a repressor. And so that repressor then has a specific binding site to bind to the operator, and it blocks RNA polymerase from binding and transcribing the genes. So then the genes are off. So there's three ways really to control operon transcription. One is to, if it's a gene that's an inducible, meaning that it's off normally, like we just looked at, a repressor protein binds to that and stops it. Another way is with a repressible operon that's normally on, a repressor can turn it off. And then the last thing we're going to look at is how they're regulated by activator proteins. So I think of these here as on switches, these here as off switches, and these here as volume control. So we're going to look at each of these. Okay, so the repressor protein has two binding sites. One is going to be where it binds to the operator, and the other is where it binds to the inducer. So here's the inducer. So when it binds, it's going to change the shape of the repressor and either allow it to bind to the operator or not allow it to bind to the operator. And if it's bound to the operator, the RNA polymerase is not going to be able to transcribe the gene, and if it's not bound, then it can transcribe the gene. And so this is our lac operon, and in this case, we have lactose present. So now the lactose itself is actually going to bind to the repressor, change the shape of it so that it can no longer bind to the operator. RNA polymerase can transcribe the gene, and the gene is coding for 3 protein products that break down the lactose, which started the whole process. So when there's no lactose, it looks like this. It binds and it blocks. When there is lactose, it changes the shape of the repressor so it can no longer bind, and then the RNA polymerase can transcribe the gene. Now, that's an inducible system, but we also have repressible systems. So this is the opposite. So here's a system that's normally on. So the lactose is not normally on unless there's lactose present. Here's a process where generally when we see this, we see something that it's like an anabolic pathway, something that the cell is making. So in this case, it's tryptophan. So the bacteria cells need tryptophan. So if they're producing lots and lots and lots of tryptophan builds up. They want to be able to shut this down. So the accumulation of the tryptophan is actually going to act as a co-repressor. So here again, we have our regulatory genes which make our repressor. But in this case, our repressor is made in a form that cannot bind to the operator. However, it can bind to the operator if a co-repressor binds to it and changes its shape. So in this case, we have an accumulation of this tryptophan, some kind of product the cell is making. As it accumulates, it binds to the repressor, changing the shape of it, allowing it to bind to the operator, and shutting down the transcription. We don't need any more of it. We've already made enough of it. So that's an off switch. And then the last, I think of this as volume control. So there are also activator proteins. And so here we have a molecule called CRP. And when CAMP, we've met this before, binds to CRP, it allows the CRP to bind to the promoter, and it actually enhances the binding of the RNA polymerase. So ultimately, you get an enhanced transcription. So you get more and more gene product. So this is sort of like a volume, increasing the volume of this. However, and this would be a situation where there's a lot of lactose and very little glucose. So we want to increase the lac operon. We want more of the genes that are going to be used to make the enzymes to break down the lactose. But then, if there's a lot of glucose present and some lactose, the cell would rather use the glucose. So remember here we have the CRP, gets bind with the CAMP, binds to the promoter, and enhances this process. Here, the CRP does not bind to CAMP, and therefore the RNA polymerase, it can bind and transcribe these genes, but it's going to be turning down the volume. We're going to be making less of that. So it's just sort of a way for the bacteria to kind of control which food sources it's using. So which pathways are more efficient for it. Okay, so as sort of an overview of what we've just done, there are repressible and inducible systems. And if you think about a repressible system is being used for anabolic pathways, or pathways where you're building something. So you need that. You don't worry about your starting material. You're trying to make something, but as that product accumulates, you slow down the process of making that, or stop the process of making it, because you don't need any more. So it's controlled by how much product you have. So you can look at those as off switches. Then there are inducible pathways, and this is a catabolic pathway where you're breaking something down. Well, clearly, if you don't have that substance to break down, you don't need that pathway to go. So that pathway is going to be controlled by how much start material there is. And so if there's start material around, then the on switch. And then again, you can have this on switch on high volume, or this on switch on a lower volume, depending on those activator proteins. And so that, I hope, helps you understand gene expression in viruses and prokaryotes. In eukaryotes, it's a much more complex system, and that's next lecture. Thank you.